Plant breeding. Within each seed lies the genetic information that determines its yield potential, how well it might resist pests and diseases, and how well it might adapt to adverse environmental conditions. Farmers expect quality seed to produce high yields. Seed quality depends, in part, on two factors: viability and vigor. Viability is a measure of how many seeds in a quantity are alive and could germinate and develop into plants under ideal conditions. Vigor measures the ability of those seeds to germinate and produce healthy seedlings under less than ideal conditions. Seed developers can improve the viability and vigor of their seeds through science. By using plant breeding methods such as selection, hybridization, and genetic engineering, plant breeders can produce varieties of plants called cultivars that exhibit optimal qualities. Selection is the oldest method of plant breeding. The best milk producer, the juiciest apple, and the most beautiful flower have been selected for breeding for thousands of years. Selection involves selecting plants with the most desirable traits, or alleles, to be parents for the next generation. If selection continues through multiple generations of offspring, the desirable trait becomes more common. Selective breeding increases the frequency of a desired allele, such as flower petal color, within a particular breeding population. Selection is often used to produce ornamental plants that come in different colors. Mutations are frequently used in the process of selection. Mutations can be harmful, but some are beneficial to the organism or to the plant breeder. If an apple tree comes from a variety that has always produced only green apples and it suddenly produces red apples, then a mutation has occurred in the green-producing variety. The offspring from the mutant tree will also produce red apples, a trait that might be desirable to a commercial fruit producer. Hybridization is the process of breeding individual plants from different varieties in order to produce an offspring that has better characteristics than either individual parent. Plants are hybridized for different types of improvements. Breeders might want to increase the amount of consumable seeds in a cold-hardy plant, or they might want to increase the cold hardiness of a particularly fragrant flower. A plant breeder might select one parent from a corn variety that produces the juicy golden kernels that consumers desire, and breed it with another parent from a corn variety that is naturally resistant to leaf blight. The breeder controls pollination for several generations. With corn, this means detasseling chosen female plants so that the tassel on the chosen male plant can pollinate the ear silks of both plants. The breeder will eventually produce a line of offspring that always show the desired characteristics of appearance and disease resistance. When offspring have better yield, hardiness, or disease resistance, breeders have achieved what is called hybrid vigor, but only in the first generation. The corn that hybridization yields, whether feed grade or food grade, is considered sterile. The seed loses its vigor over the next 2 to 3 generations and is not worth the cost of planting. Commercial producers no longer select the best ears of corn and save them for the next year's planting. With hybridization, they purchase seed corn and any other crop seed for planting each year. Today, about 99% of all corn grown in the United States is grown from hybrid seed. Genetic engineering is different from selection and hybridization because it goes beyond trying to manipulate gene combinations within a species over time. Genetic engineering is the process of deliberately altering the genetic makeup of an organism, and it's faster and more reliable than breeding methods. Scientists who study genes are called geneticists. Geneticists can use recombinant DNA technology to take genes from an organism, such as bacterium, and combine them with the DNA of a plant. For example, bacteria called Bacillus thuringiensis naturally produce a virulent toxin that is deadly to various insects, including moths, mosquitoes, and beetles. This is what the BT designation means in BT corn or BT cotton. Geneticists are able to isolate genes in the bacteria's DNA that allow the bacteria to produce the toxin. They snip these gene fragments from the bacteria's DNA, and insert them into the DNA of a plant, like cotton, corn, or potatoes. The newly inserted genes combine with the plant's DNA and allow the plant to produce the same toxins that the bacteria produce. When insects feed on the leaves of the genetically modified plant, they eat the newly produced poison and die. Using genetic engineering, scientists are able to make crops resistant to insects, diseases, viruses, and weed killers, and even increase their nutritional value and climate hardiness. And now, let's explore the core. A mutation is any change in the DNA sequence of a gene. It's a naturally occurring variation that cannot be predicted. If a mutation occurs in either a male or female sex cell that takes part in fertilization, then the mutation is passed on to the offspring. Mutations can be caused by external agents, called mutagens, such as exposure to high temperatures, radiation, or certain chemicals that damage genes or break apart DNA. Mutations can also be caused by errors that occur spontaneously within the cell during DNA replication, transcription, and cell division. RNA molecules use instructions from DNA to build proteins. If an error occurs in translating the instructions, then two types of mutations can occur. This sentence shows what happens in a point mutation. Changing only one letter in this sentence changes one word, and it alters the meaning of the sentence. A point mutation is a change in a single base pair of the DNA. Notice how changing only one base changes the codon, causing a different amino acid to be inserted into the chain. This could interfere with how the protein functions. Another error in translation, called a frameshift mutation, could also occur. This sentence shows what happens in a frameshift mutation. Deleting the G changes every word in the rest of the sentence. In a frameshift mutation, a single base is either added or deleted. Notice how this shifts the reading of every codon that comes after the mutation, and changes almost every amino acid that follows in the protein chain. You can see how frameshift mutations tend to cause more genetic change than point mutations. Currently, geneticists are working on targeted mutation. With great precision, they are trying to create errors in the DNA of crop plants that would lead to desirable mutations. This type of genetic modification might be more desirable to consumers who don't like the idea of eating corn that contains foreign DNA from a bacterium. Today's SAT word is virulent. Virulent is an adjective that means violently hostile or actively poisonous. In the lesson, we learned that DNA from bacteria producing a virulent or actively poisonous toxin can be inserted into crop plants to make them toxic to specific insect pests.